interesting fungi. Last time we went through some interesting stuff. We talked about how it kind of grows and expands out, and I found it really interesting that the fungi isn't a plant or an animal, but it's its own separate thing. And I find it weird that, when it's at the little spore stage and it's swimming around, I wonder how much control of its direction it has when it swims. Like how intelligent is it actually? Seemingly more intelligent than plants. Anyway, here we are for part 2. If you have anything you want me to whisper about, please tell me in the comments below. I read every comment and I'm always happy to do a video for you. Now, you may hear in the background it started raining. The rain's pouring down a little bit. I hope that's not too disturbing for you. Now we are moving to the interesting reproduction of the fungi. Please remember that I really can't say scientific names, so I'm going to be slaughtering it in this video. Okay, now on to reproduction. Fungal reproduction is complex, reflecting the differences in lifestyles and genetic makeup with this diverse kingdom of organisms. It is estimated that a third of all fungi reproduce using more than one method of propagation. For example, reproduction may occur in 2 well-differentiated stages within the life cycle of a species: the teleomorph and the anamorph. Environmental conditions trigger genetically determined developmental states that lead to the creation of specialized structures for sexual or asexual reproduction. These structures aid reproduction by efficiently dispersing spores or spore-containing propagules. Moving to asexual reproduction. Asexual reproduction occurs via vegetative spores, conidia, or through mycelial fragmentation. Mycelial fragmentation occurs when a fungal mycelium separates into pieces, and each component grows into a separate mycelium. Mycelial fragmentation and vegetative spores maintain a clonal populations adapted to a specific niche, and allow more rapid dispersal than asexual reproduction. The fungi imperfecti, fungi lacking the perfect or sexual stage, or Deuteromycota, comprise all the species that lack an observable sexual cycle. Deuteromycota is not an accepted taxonomic clade and is now taken to mean simply fungi that lack a known sexual stage. Now we move to sexual reproduction. Sexual reproduction with meiosis has been directly observed in all fungal phyla except Glomeromycota. However, genetic analysis suggests meiosis in Glomeromycota as well. It differs in many aspects from sexual reproduction in animals or in plants. Differences also exist between fungal groups and can be used to discriminate species by morphological differences in sexual structures and reproductive strategies. Mating experiments between fungal isolates may identify species on the basis of biological species concepts. The major fungal groupings have initially been delineated based on the morphology of their sexual structures and spores. For example, the spore-containing structures Acci and Basidia can be used in the identification of asomocytes and basidiomycetes, respectively. Fungi employ 2 mating systems. Heterothallic species allow mating only between individuals of opposite mating types, whereas homothallic species can mate and sexually reproduce with any other individual or itself. Most fungi have both a haploid and diploid stage in their life cycles. meiosis is required for the initiation of the sexual cycle. Many escapisms and basidiomycetes go through a dikaryotic stage in which the nuclei inherited from the two parents do not combine immediately after cell fusion, but remain separate in the hyphal cells. In the asomycetes, dikaryotic hyphae of the hymeneum, the spore-bearing tissue layer, form a characteristic hook at the hyphal septum. During cell division, formation of the hook ensures proper distribution of the newly divided nuclei into the apical and basal hyphal compartments. An ascus, which is plural for asci, is then formed, in which karyogamy, which is nuclear fusion, occurs. Asci are embedded in an asocarp or fruiting body. Karyogamy in the asci is followed immediately by meiosis and the production of ascophores. After dispersal, the ascospores may germinate and form a new haploid mycelium. Sexual reproduction in basidiomycetes is similar to that of asomycetes. Compatible haploid hyphae fuse to produce a dikaryotic mycelium. However, the dikaryotic phase is more extensive in the basidiomycetes, often also present in the vegetatively grown mycelium. A specialized anatomical structure called a clamp connection is formed at each hyphal septum. As with the structurally similar hook in the asomycetes, the clamp connection to the basidiomycetes is required for controlled transfer of nuclei during cell division. To maintain the dikaryotic stage with two genetically different nuclei in each hyphal compartment, a basidiocarp is formed in which club-like structures known as basidiae generate haploid basidiospores after karyogamy and meiosis. The most commonly known basidiocarps are mushrooms, but they may also take other forms. See morphology section. In fungi, formerly classified as zygomycota, haploid hyphae of two individuals fuse, forming a gametangium, a specialized cell structure that becomes a fertile gamete-producing cell. The gametangium develops into a zygospore, a thick-walled spore formed by the union of gametes. When the zygospore germinates, it undergoes meiosis, generating new haploid hyphae, which may then form asexual sporangiospores. These sporangiospores allow the fungus to rapidly disperse and germinate into new genetically identical haploid fungal mycelia. Now moving to spore dispersal. Both asexual and sexual spores or sporangiospores are often actively dispersed by forcible ejection from their reproductive structures. This ejection ensures exit of the spores from the reproductive structures as well as travelling through the air over long distances. Specialized mechanical and physiological mechanisms as well as spore surface structures such as hydrophobins enable efficient spore ejection. For example, the structure of the spore-bearing cells in some asomycete species is such that the buildup of substances affecting cell volume and fluid balance enables the explosive discharge of spores into the air. The forcible discharge of single spores, termed bellistospores, involves the formation of a small drop of water, the bullous drop, which upon contact with the spore leads to its projectile release with an initial acceleration of more than 10,000 g. The net result is that the spore is ejected 0.01 to 0.02 centimeters, sufficient distance for it to fall through the gills or pores into the air below. Other fungi, like the puffballs, rely on alternative mechanisms for spore release, such as external mechanical forces. The hydnoid fungi, the tooth fungi, produce spores on pendant tooth-like or spine-like projections. The bird's nest fungi use the force of falling water droplets to liberate the spores from cup-shaped fruiting bodies. Another strategy is seen in the stinkhorns, a group of fungi with lively colours and putrid odour that attract insects to disperse their spores. Most of the researched species of fungus are transported by wind. Such species often produce dry or hydrophobic spores which do not absorb water and are readily scattered by raindrops, for example. Moving to homothalism. In homothallic sexual reproduction, the two haploid nuclei derived from the same individual fuse to form a zygote that can undergo meiosis. Homothallic fungi include species with an aspergillus-like asexual stage, animals occurring in numerous different genera. Several species of the Asomycete genus Cochleobolus and the Asomycete Pneumocystis girovexi Heitman reviewed evidence bearing on the evolution of sexual reproduction in the fungi and concluded that the earliest mode of sexual reproduction among eukaryotes was likely homothalism, that is, self-fertile unisexual reproduction. Now moving to other sexual processes. Besides regular sexual reproduction with meiosis, certain fungi, such as those in the genera Penicillium and Aspergillus, may exchange genetic material via parasexual processes initiated by anastomosis between hyphae and plasmogony of fungal cells. The frequency and relative importance of parasexual events is unclear and may be lower than other sexual processes. It is known to play a role in intraspecific hybridization and is likely required for hybridization between species, which has been associated with major events in fungal evolution. Now moving to evolution again. In contrast to plants and animals, the early fossil record of the fungi is meagre. Factors that likely contribute to the underrepresentation of fungal species among fossils include the nature of fungal fruiting bodies, which are soft, fleshy, and easily degradable tissues, and the microscopic dimensions of most fungal structures, which therefore are not readily evident. Fungal fossils are difficult to distinguish from those of other microbes, and are most easily identified when they resemble extant fungi, often recovered from a permineralized plant or animal host. These samples are typically studied by making thin section preparations that can be examined with light microscopy or transmission electron microscopy. Researchers study compression fossils by dissolving the surrounding matrix with acid and then using light or scanning electron microscopy to examine surface details. The earliest fossil possessing features typical of fungi date to the Paleoproterozoic era, some 2,400 million years ago. These multicellular benthic organisms had filamentous structures capable of anastomosis. Other studies, e.g. one in 2009, estimate that the arrival of fungal organisms at about 760 to 160 million years ago on the basis of comparisons of the rate of evolution in closely related groups. For much of the Paleozoic era, from 542 to 251 million years ago, the fungi appear to have been aquatic and consisted of necessitated a diversification of ecological strategies for obtaining nutrients, including parasitism, saprobubism, and the development of mutualistic relations such as mycorrhiza and lichenization. Recent 2009 studies suggest that the ancestral ecological state of the Ascomycota was saprobism, and that the independent lichenization events have occurred multiple times. In May of 2019, scientists reported that the discovery of a fossilized fungus named Urosphera giraldia in the Canadian Arctic may have grown on land 1 billion years ago, well before plants were living on land. Earlier, it had been presumed that the fungi colonized the land during the Cambrian explosion, which is also long before land plants. Fossilized hyphae and spores recovered from the Ordovician of Wisconsin, which is 460 MA, resemble modern-day glomeruli and existed at a time when the land flora likely consisted of only non-vascular birophyte-like plants. Prototaxites, which was probably a fungus or lichen, would have been the tallest organism of the late Silurian and early Devonian. Fungal fossils do not become common and uncontroversial until the early Devonian, which is 460 to 359 MA, when they occur abundantly in the Rhynie chert, mostly as Zygomatica and Chytridiomycota. At about this same time, approximately 400 MA, the Ascomycota and Basidiomycota diverged, and all modern classes of fungi were present by the late Carboniferous. Lichen-like fossils have been found in the Dushanto Formation in southern China, dating back to 635 to 554 MA. Lichens form a component of the early terrestrial ecosystems, and the estimated age of the oldest terrestrial lichen fossil is 400 MA. This date corresponds to the age of the oldest known sporocarp fossil, a Paleopyrenomycetes species found in the Rhine chert. The oldest fossil with microscopic features resembling modern-day Basidiomycetes is Paleoancestros, found permineralized with a fern from the Paleocene. Rare in the fossil record are the Homo Basidiomycetes, a taxon roughly equivalent to the mushroom-producing species of the Agariosemys. Two amber-preserved specimens provide evidence that the earliest known mushroom-forming fungi, the extinct species Archaeomycemius legati, appeared during the late Cretaceous. Sometimes after the Permian-Triassic extinction event, a fungal spike, originally thought to be an extraordinary abundance of fungal spores in sediments, formed, suggesting that fungi were the dominant life form at this time, representing nearly 100% of the available fossil record for this period. However, the relative proportion of fungal spores relative to spores formed by algal species is difficult to assess. The spike did not appear worldwide, and in many places it did not fall on the Permian-Triassic boundary. 65 million years ago, immediately after the Cretaceous Paleogene extinction event that famously killed off most dinosaurs, there is a dramatic increase in evidence of fungi. Apparently the death of most plant and animal species, leading to a huge fungal bloom like a massive compost heap. Wow. Now we move to the taxonomy. Although commonly included in botany curricula and textbooks, fungi are more closely related to animals than to plants, and are placed with the animals in the monophyletic group of Opisthokonts. Analysis using molecular phylogenetics support a monophyletic origin of fungi. The taxonomy of fungi is in a state of constant flux, especially due to recent research based on DNA comparisons. These current phylogenetic analyses often overturn classifications based on older and sometimes less discriminative methods based on morphological features and biological species, concepts obtained from experimental matings. There is no unique, generally accepted system at the higher taxonomic levels, and there are frequent name changes at every level, from species upwards. Efforts among researchers are now underway to establish and encourage usage of a unified and more consistent nomenclature. Fungal species can also have multiple scientific names depending on their life cycles and mode, being sexual or asexual, of reproduction. Websites such as Index, Fungorum, and IDIS list current names of fungal species with cross-references to older synonyms. The 2007 classification of kingdom fungi is the result of a large-scale collaborative research effort involving dozens of mycologists and other scientists working on a fungal taxonomy. It recognizes 7 phyla, 2 of which, the Ascomycota and the Basidomycota, are contained within a branching representation subkingdom Dicaria. The most species-rich and familiar group, including all the mushrooms, most food spoilage molds, most plant pathogenic fungi, and the beer, wine, and bread yeasts. Okay, my dear friends, that was super interesting, but I do have to end it here, as I do have a finite amount of time. Now, there's probably about 1 more episode's worth of Wikipedia about the fungi. Please tell me in the comments below if you would like the part 3 for the rest of the Wikipedia page. I may revisit the rest of the Wikipedia in the future if you are actually interested. I know that this is getting very scientific because this is, of course, its own, branch, its own scientific kingdom, so it makes sense that it gets a bit scientific. But I found it really interesting so far, and I hope you liked this video. Now, tell me in the comments below if you want a part 3 or if you want a different Whispering Wikipedia